Okay, cool. The record is on. So hello and welcome everyone. So welcome back to the MSA SPS, or the Mycological Society of America's student and postdoc section. So this is the Students' Mycological Symposium. We'll be starting on the dot to give our main speakers enough time to explain their work and for questions. And we're excited to have you here for our 5th meeting, okay? My name is Tanya Kurbasoyan. I am a 4th year microbiology graduate student in Dr. Jason Steich's lab, and I'm the vice chair of the MSA SPS. So hopefully I'll have other members of the executive board coming in soon so I can introduce them, but probably later on at the end. So welcome. Firstly, I'd like to say if you have any questions, please write them down in the chat and we can address it later. And either I can read it to the speakers or I can call upon you and you can unmute yourself and ask. This meeting, as I mentioned earlier, will be recorded and will be available on YouTube. So that way we'll share it on Facebook and Twitter as well. The talks will generally be around 20 to 25 minutes long, and this will leave the speakers about 5 minutes for questions, thus filling in the hour. So we have 2 awesome speakers, 2 Lycan ladies joining us today. And first we'll start with Gulnara. So Gulnara is a PhD candidate at the Spribilli lab at the University of Alberta. She studies the evolution of fungal algal symbiosis, known as lichens. In her project, she is trying to balance system biology, symbiosis studies, fungal biology, and bioinformatics. So prior to her PhD, she received her bachelor's and master's from St. Petersburg University in Russia, where she studied conservation biology of lichens. So Gulnara, welcome to begin. Share your screen.
Thank you. Okay. Okay, so now hopefully I can see my screen. Hello, everyone. I'm really excited about presenting my research to you today. So yeah, on this slide, you can see my name and my Twitter profile. If you want to connect with me, I would love to. But now let me just start. And I called my talk Metagenomics of Lichen Symbiosis and Nailing Down Unculturable Fungi. So according to some estimates, about 90% of fungi are unculturable. So the fungi that we cannot grow in a lab. And in addition to being a lion's share of fungal diversity, many of these fungi play important roles in microbial communities, but studying them is complicated. A usual alternative to study unculturable lineages is metagenomics. And metagenomics is the study of sequence data from intact environmental samples. And metagenomics works really well for prokaryotes, but applying it to fungi can be challenging. So first, fungal genomes are much, much larger than bacterial genomes, and that already can cause problems. Also, some fungi have a lot of going on with ploidy situation, and you have like multiploid cells and whatnot. And lastly, fungal genomes can be heterogeneous due to presence of transposable elements or other mobile DNA elements. And all these factors can really mess with bioinformatic algorithms that metagenomics relies upon. And eventually, it means that applying to fungi techniques that work really well for bacteria can be complicated. And that's why fungi and fungal communities are often underreported and understudied. Sorry. Lichen symbiosis can be seen as a case of microbial community where fungi play crucial roles. Lichen symbiosis consists of multiple lineages. One of them is the so-called dominant fungus. Typically, it is an ascomycete in its hyphal form. Then there are unicellular phototrophs, typically green algae or cyanobacteria. And in addition to that, there is a suite of low abundance symbionts that includes both yeasts and bacteria. Together, all of the symbionts can form a lichen. And lichen is a symbiotic association that is very stable. It has its own really well-recognizable morphology and body plan. It can play a role in ecosystem and so on. By the way, the lichen that you see on the screen is called Alectoria sarmentosa. Its common name is witch's hair lichen, and this is our model system for this study. So how does lichen symbiosis work? The current understanding is that the alga produces sugars that it delivers to the fungus. And the dominant fungus, in turn, creates the symbiotic body to host these algal cells. But no one really knows how the low abundance partners fit into that picture. What is their relationship to the dominant partners? What do they take from them, and do they contribute anything to the lichen symbiosis? All of these questions are largely unknown. There has been several hypotheses made that these partners are mutualistic symbionts or that they are parasites, but until recently, we really didn't know one way or the other. And one of the reasons why is that lichens are extremely hard to study experimentally. So the dominant fungus can be cultured and grown in a lab, but it grows extremely slowly. So when I say slowly, the picture that you see on the slide, it took us about 1 year to grow this culture so big. And the yeasts are even worse in some ways. The yeasts are unculturable, at least as of now. And that means that we cannot really study their role in the symbiosis in any experiment. Also, applying metagenomics to them is hard, partially because of the challenges that I mentioned before, like large complex genomes, but partially because in lichens, they are present in really low abundance. And that factors together made yeast into some sort of enigma. Like, we know that they are present, but really we don't know much else about them. One of the goals for the study was to overcome the challenges and obtain the genomes of the lichen yeasts. To solve the problem of low abundance, we used a method that we called Cortex-Slurry Method. To explain what it is and why we did what we did, let's us first talk a little bit about lichen and how they look on a microscopic level. So on this slide, you see the scanning electron microscopy image of a cross-section through a lichen thallus. First thing that you notice is that there are two types of cells. The green blobs are the algal cells, and the long spaghetti-like cells are the hyphae of the dominant fungus. But I wanted to draw your attention to this layer that in lichens is called cortex. Cortex is the outermost layer of lichens, and on this picture, it kind of looks like a pie crust on a lichen. And this pie crust is really important for lichen symbiosis because this is basically the main structural agent. The cortex shapes lichens and gives them all of this very recognizable body plans. But more importantly now, the yeasts are embedded in this layer. So the layer consists of the cells of the symbionts that are glued together by a thick layer of extracellular matrix, and the yeasts are embedded in this matrix. So we decided to dissolve the matrix, and to do that, we took a lichen sample and they put it in hot water. Then they put that hot water into a shaker for several hours, and that setup was really a little bit like a washing machine kind of thing. Then we concentrated the solution that contained this yeast cells released, and this sample that we got, we called it Cortex-Slurry. And then we extracted DNA from it and sequenced it to make a metagenome. So with the problem of low abundance out the way, the next step was metagenomic analysis. This analysis included 3 steps. The first was assembly. And the metagenomic data that we produced, we got from an Illumina sequencing machine. And that means that we got a lot and lots of really short sequences, about 250 base pair longs. And the goal of this step is to assemble these short pieces into larger contiguous sequences. Sometimes people use a metaphor and say that this process is a little bit like assembling a jigsaw puzzle. The second step is binning. The goal of this step is to disentangle the metagenomic data and extract genomes of individual organisms. So if I want to extend the metaphor one step further, I would say that metagenomics is a little bit like dealing with a situation where you have a dozen of jigsaw puzzles all mixed together in one box, and you somehow have to deal with it. And I just wanted to share this piece of art from a Canadian artists. Yeah, basically, when you do metagenomics, you have to try really hard to avoid ending up with a situation like that. Okay, so how we did it, how we extracted individual genomes. We used a database-independent approach. This means that we didn't try to identify genomes by comparing our data to already published sequences. This approach would not work because it only allows to obtain genomes that are similar to the ones that you already have. And because we started with no genomes available to us, that really wasn't an option. Instead, we relied on trends and patterns within our data. And that included two main things. The first was coverage, and coverage is a proxy of cell abundance. The idea is that if one organism have more cells in your sample, more copies of a genome will be sequenced in the metagenomic data. And you can use this information to separate high abundance genomes from low abundance genomes. The second thing is sequence content. And sequence content basically means the difference in distributions of oligonucleotide frequencies. And just one example to explain what this means is that you can look at patterns of how often nucleotide sequences occur in a genome. And apparently, because of how genome evolution is happening, one genome can have, for example, more ACT sequences within its genome than AGT sequences. And the other genome might have it the other way around. And you collect all of this data and you analyze it, and this can be enough to separate genomes from different organisms. So, and after you do that, the final step is quality control. You have to make sure that the genomes are, the genomes that you produced are of a good quality. Okay, so that was a bit of a lengthy introduction, and now we can finally get to the part where we see what we find out. So, first thing that I wanted to mention is that we found two genomes in our metagenome that we didn't expect to find. In total, we were able to extract 4 genomes. The first belonged to the dominant fungus. The second belonged to Cyphobasidium. And Cyphobasidium is a species of lichen yeast, and we kind of expected to find it because it was reported from our lichen before. Tremella is also another yeast that we didn't know occurred in our lichen, so that was new. Tremella includes a lot of lichen-associated fungi. Until recently, all of them were thought to be pathogenic, but now we know that Tremella in its yeast form is absolutely ubiquitous in a different species of lichen, in Litharia lichens. Also, Tremella is related to cumin pathogen Cryptococcus neoformans. And finally, the fourth metagenome wasn't a fungal. The fourth genome wasn't a fungal, it was a genome of Granulicella bacterium. Okay, so after we found all of these genomes, the next step that we had to take was to establish whether these organisms that we identified are present in our lichen consistently, because there also was a possibility that they are just like random contaminants from the environment. We had to rule it out. And to do that, we did PCR screening. So this is a diagram to show the results of the screening. I hid the data for now so I can explain the figure, and then I will show you the data. So what we did, we went to 3 different locations in Canada, and in each location we sampled, we selected several trees and we collected samples. From each tree, we collected 3 samples. One of them was Alectoria, the lichen that we study. The second was any other lichen that grows in vicinity. And the third specimen was just bare tree bark. And we wanted to know 2 things. First, if the organisms that we identified in our metagenomes, if they are present in Alectoria consistently, are they always there, or is it more like a hit and miss situation? And also, we wanted to know if these organisms are specific to Alectoria, because as I said, we wanted to rule out the possibility that they are just present in the environment and like present in the tree bark and everywhere. So we needed to exclude that. And this is what we found. If you look at this row with Alectoria samples, you see that Cyphobasidium and Tremella occur in vast majority of Alectoria samples. And in every Alectoria specimen, there was at least one of those yeasts. With the bacterium Grunellicella, the situation was kind of the reverse. It almost never occurred in Alectoria, but it was quite common in tree bark. So we concluded that Tremella and Cyphobasidium are consistently present in the symbiosis, and that Grunellicella is not. And that's why we didn't really look much further into this bacterium. Okay, so that was a little bit of like a side quest. And now we can get back to the genomic data. So our final data set included 3 fungal genomes, the dominant fungus and 2 yeasts, Cyphobasidium and Tremella. Each of these genomes had high quality, comparable to what you can expect if you sequence a genome from culture. We annotated the genomes, meaning that we predicted genes present there, and each gene we assigned with a putative function. Okay, so what we can tell about the biology of this fungi based on the genomes. We looked into many aspects of these genomes, but for now, I wanted to share with you 3 highlights. And for each, I'm going to first give a little bit of background to explain why we became interested in this thing to begin with, and then I will share with you the results. And the first is secondary metabolism. Lichens are extremely rich in secondary metabolites, and secondary metabolites, for context, are various specialized compounds that include hormones, pigments, antibiotics, and such things. And I'm really looking forward to Susan Stock, who will tell us more about lichens and secondary metabolites. But another thing that is more relevant to my study is that in one case, the concentration of a secondary metabolite has been linked to abundance of lichen yeasts. So this secondary metabolite is called Vulpinic acid, and Vulpinic acid is like very bright yellow pigment. And so here is a figure from another paper from our lab. And it shows lichen samples on the left, and on the right, you see highlighted with green are the yeast cells. And you can see that the lichen sample that is brown and doesn't contain much of vulpinic acid also has comparatively few yeast cells. And the more yellow sample with a lot of vulpinic acid has more yeasts. So the hypothesis has been made that the yeast might be involved in secondary metabolism production. And this is what we found. On this bar graph, you see number of secondary metabolism gene clusters in different genomes. And the genome of the dominant fungus of Alectoria here had way more of these genes compared to both Cyphobasidium and Tremella. And we can hypothesize that probably in this lichen, the dominant fungus is most responsible for production of secondary metabolites. I want to put an asterisk here and say that the Lichen Symbiosis that we used was different from the one I showed you on the picture before, and it's entirely possible that in different Lichen Symbiosis, it works a different way. Okay, next. class of enzymes that we looked into were carbohydrate-active enzymes, or Caseins. These are proteins that are involved in producing, modifying, and breaking down polysaccharides. And we became interested in these Caseins because polysaccharides play a lot of important roles in lichens. So if you remember the picture from before, and this cortex layer that I mentioned, the extracellular matrix of cortex is mostly made from polysaccharides. So polysaccharides are the substances that glue the cells into a lichen shape, and also the yeasts are embedded in these polysaccharides. So that's why we wanted to know more about them. And here you see the casein profile of the three fungi, and there's a lot to talk about different caseins, and I could probably make like an entire talk about them, but I wanted to focus on one example. Both yeasts, but not the dominant fungus, had genes responsible for synthesis of GXM, or glucuronoxylomannan. Glucuronoxylomannan is a polysaccharide that is known from non-lichen relatives of Tremella. So for example, in Cryptococcus, the human pathogen, GXM forms a capsule that helps the fungus to evade human immune response. Also, GXM is extremely hygroscopic. So on this slide, you can see Tremella fuciformis, another relative, and Tremella fuciformis has a common name, which is jelly fungus. And this is exactly because of this jelly-like texture that the polysaccharide gives it. And moisture-retaining properties of GXM are even used in cosmetics, like on the slide you see a skin lotion made from Tremella. So we hypothesized that maybe the yeasts produce GXM in lichens too, and that helps lichen to retain water. Okay, and finally, one of the questions that we hoped to answer was the nature of relationship between the yeast and the dominant fungus. So as I mentioned, several hypotheses has been made that they are parasites, and this was largely based on the fact that this fungi very rarely can cause gall-like deformations, like the one that you see on the slide. So many people assume that this means that this fungi are always pathogenic. But sort of against that hypothesis spoke the fact that the yeasts are absolutely ubiquitous even in healthy looking specimens. So they probably don't cause harm usually. But either way, it was something that was debated, and we really hoped that our genomes will help us to answer this question. But it turned out that genomic profiles were not particularly helpful here. First, because we don't know much about fungal relationships compared to, for example, how much we know about fungal plant relationships. And that means that there wasn't enough point of comparison for us. Also, as we know from plant-associated fungi, the same protein sometimes can be used in both mutualistic and parasitic contexts, and that means that genomic profiles can be really similar, even when the relationships are vastly different. And also, to that matter, mutualism and parasitism is almost always a little bit of a spectrum. So we were not able to answer that question, but there is some evidence that I wanted to mention. So first, Tremella has a profile of a nitrogen oxytroph, and that means that it cannot assimilate nitrates or nitrates from the environment. It has to get organic nitrogen from some other living organism. Second, all three fungi have putative polyol transporters. And why is it important? Well, because the sugar that the alga delivers to the dominant fungus is a polyol. So, and now we know that the yeast have transporters of polyols too. And finally, Cyphobasidium, based on its genome profile, can produce enzymes that specifically target plant polysaccharides. So taken together, this can be viewed as evidence that the yeast might rely on alga for sugars. And that can be as evidence against the hypothesis that the yeast are parasitic on the dominant fungus. Okay, so to sum up, we used a combination of a wet lab technique we called Cortex-Slurry with bioinformatic analysis to obtain the first two genomes of lichen yeasts. When we started our study, basically what we knew about lichen yeast was that they exist and their taxonomy. so what fungal group they belong to. But now with these genomes, we were able to generate a set of hypotheses about the metabolic potential of the yeast and the trophic strategies. And this is the first ever evidence on the role the yeast might be playing in Lichen Symbiosis. And with that, I wanted to thank all collaborators from Spribbilla Lab, from EBI, EMBL, and Agriculture and Agri-Food Canada, thanks to the funding sources. And the last thing that I wanted to mention is that our lab at the University of Alberta is hiring. There are 3 positions, 1 postdoc and 2 PhD students, and we're just a few days before the deadline. You can learn more on our lab website, and if you want to ask me any questions about our lab, about our university, you are welcome to contact me on Twitter or by email. And with that, I wanted to thank again the organizers and thank you for your attention.
Awesome. Thank you. Wow, it looks like we got a couple of questions here. Erin, I see you posted a question. Did you want to ask it yourself or would you like me to read it?
I can ask it.
Okay.
So have you guys tried to compare the genomes of the yeast and the lichen forming fungi and like try to figure out if there's any like really active genes that are activated during the lichen formation, such as like melanin production or any secondary metabolites for like fungus on fungus wars or anything like that? I don't know if you guys have done that yet or not.
Yeah, so we tried to compare the yeast and the like dominant fungus, as I call it, genomes. And in general, they're kind of different. Like the dominant fungi genomes are much larger to begin with. Yeah, so secondary metabolites that, yeah, we tried to look into that, and in the yeast genomes there's not much going on in terms of like this warfare between fungi. We tried to look at every like possible like toxin or something like that, and it turns out that actually the dominant fungus seems to be able to produce toxins and the yeasts are not really. And if you look at the so-called effectors, like the genes that are known to be associated with pathogenicity and like suppressing host immune response and all of that, the dominant fungus seems to have more of those than the yeast. But actually we kind of expected the reverse, but yeah. But at the same time, like again, it's really hard to draw like big biological conclusions based on that, because yeah, we don't know if those genes are expressed, like what is the situation there. We tried to like a little bit leverage comparing the genome of lichen yeast to closely related genomes that are not associated with fungi, and yeah, there was like a little bit of signal there, but yeah, it's kind of hard to separate this like phylogeny component in any difference.
Awesome. Thank you.
Awesome.
Also, it's nice to see you.
Awesome. Thank you. I think we have time for one more question. I see Jessica Bernardin asked, I don't know if you'd like to ask the question, if you want me to read it.
Sure. First off, great talk. Thank you so much. I love lichen. I love hearing about it, and it's exciting to see so many people interested in it. I just wanted to know if you thought, just your opinion, if you thought maybe the yeast provided any benefit to the dominant fungi.
Yeah, so we try to explore all the things that, based on genomes and based on like general understanding, what the yeast might contribute to the symbiosis overall. I would say that the like benefit to the dominant fungus can be seen as a little bit like a philosophical question. What is beneficial to the dominant fungus? And I would say that if the yeasts are increasing somehow the fitness of symbiosis in general, that should be a benefit of a dominant fungus. So, and that's why we, for example, what I mentioned about this polysaccharide that we think might be beneficial for lichen, because it helps lichen to retain water, and lichens are really dependent on like being able to hold water. And that's like one hypothesis that we made. It is a hypothesis, we have to test it. But yeah, also there were some other evidence that I didn't mention in the talk about how this yeast might be involved in like nutrient scavenging or something like that too. Yeah.
Sweet. Okay, thank you, Gulnara. Thank you so much. Wonderful presentation. There are a couple more questions in the chat. So as Susan gets ready to start her presentation, maybe you can look into them and answer them there.
Sure.
Okay, cool. So Susan, let's go ahead and introduce you. So now our second speaker, Susan, she received her PharmD from University of Louisiana at Monroe College of Pharmacy in 2019. And she's like, you know what, that wasn't enough. So she's currently now a second year in her PhD program at the University of Manitoba, focusing on lichen biosynthesis. So Susan, go ahead.
Thank you for that introduction. Yes, I am a little bit crazy, but you know what, that's fine. Let me get the presentation mode real quick. Oh my gosh, my computer is being... We're getting there. Technical difficulties. Yes. Okay, so hello everyone. Like Tanya has mentioned, my name is Susan, and fun fact about this picture in the slide, it was actually for a lichen photo shoot that my boss decided, hey, I got a perfect job for you. You're gonna take a picture of lichens. So I paired up with a photographer in my department and got to take pretty pictures of lichens. So my talk today will be what do we understand about the lichen biosynthetic gene clusters? And so like Gulnara has mentioned, kind of the symbiotic relationship is mostly between the photobiont, that's generally a cyanobacteria or algal species, and the main mycobiont. And so in the middle, I just put in a structure of what a lichen might look like under the microscope. Is not the one fits all kind of structure, but it's just kind of give an example of how the algal cells are interacting with the fungal cells. And so once this relationship comes together, then we have these interesting secondary metabolites at the bottom of the screen that is specific to lichen fungi only, and you will only see it in lichen fungi. Anytime you try to see it in a non-lichen fungi, there's just something different about that chemistry that just makes it unique. And so my story kind of started a bit with learning a bit about the properties, more of the biopharmaceutical properties of lichen secondary metabolites. And so what I put on the screen are the top 3 most well-known to the lichen field. It's Arsenic acid, atrinorin, and Fiumar protocitric acid. And so these belong in the classes of dibenzofurans, depsides, and depsinones. And again, unique to lichen fungi only. Whenever you look at non-lichen fungi, they start adding like these double bonds, which makes it different. And so some of the bioactive properties, we do have the ecological aspect of like UV protection and antimicrobial properties that will protect the lichen against its environment. But additionally for humans, we've kind of saw some interest as these metabolites had some activity towards like certain cancer cell lines, and kind of a big one was like the antimicrobial, especially against MRSA. And so there's just a whole bunch of potential for these lichen metabolites being pharmaceutical drugs. So kind of what I was working research-wise in the pharmacy realm was collecting lichens in the field. I didn't personally do spot tests myself, but I know lichenologists like to do spot tests to see whether or not they're getting the correct species or not. And so I was mostly collecting not this species, but a species of Usnea. And so we were collecting just a whole bunch of them in the refugees in around the Monroe, Louisiana area. And so we extracted it. And so we end up with this crude extract. So kind of a thing that natural products chemists like to do, at least back in the day, not so much now, is we like to isolate and see what's actually in there. And so we would use different isolation techniques. My lab personally did column chromatography. That's we mostly separated based on polarity. And so from there, we were able to do structure elucidations through techniques like NMR or mass spec. And we were able to determine either known compounds or new compounds. I yet to have new, but at least we were able to find what's known. And so that's where I kind of came to the John Sorensen lab, because as I mentioned with the workflow, there was just so much time consuming and it took so much samples, but lichens grow really slow, like 1 centimeter in diameter per year, like that's slow. So we kind of decided to go a different route and see what we can do. And so my lab kind of mostly focuses on biosynthesis. And so this is basically looking at lichens and understanding how they form the compounds that we do. And so the first compound that my lab was able to make a putative identification of was the arsenic acid gene cluster. So first, we were looking at like the starter units and see what could be made. So that's how we have the structures on the left. And they go through various enzymes and then form to create this lovely structure number 2, which is methylfluoroacetophenone, which these are the starter units to make arsenic acid. But then there's also another aspect of lichens that can actually make 3-methyl-orsolinic acid, which is number 3. So lots of enzymes. We don't know where to really start. We just knew, okay, like we know X, Y, and Z enzymes are needed to make this happen. And so what we did afterwards was we kind of selected a lichen that had only arsenic acid as the metabolite. And so we picked Cladonia uncialis. It's a very popular lichen across Canada. You could just see it everywhere. You can see it in Manitoba. You can see it in Newfoundland. So it was really easy to collect a lot of it. And so my boss's first PI student, whenever she was pregnant, she actually was culturing these lichens just to get some work done while she was carrying her child. And so through that, we were actually able to get a genome of the Cladonia uncialis. And basically through process of elimination, we were able, or the lab was able to identify the arsenic acid biosynthetic gene cluster. And so we have the main enzyme making that methylfluoroacetophenone unit. And then we have the oxidative enzyme putting the two together. And so we were able to say, okay, we think this makes arsenic acid. So the next experiment to kind of determine whether or not we have the correct identification is through heterologous expression. Now, if you haven't really seen this before, that's okay. I'll explain a bit through it. But it's kind of a new technique, at least for natural products chemists, to be able to culture or to be able to get more metabolites through various host expression systems. So, and it's very cost effective in some ways, because if you put your genes of interest into, say, an E. coli's are pretty cheap to get. You can culture them in about 2 days, boom. Like you don't have to worry about going to some field to go collect some lichen. And there's actually been some success stories of people being able to get cryptic genes and silent genes to be able to work through these expression systems. And so what happens is you get your organism of source and you culture it. And so once that you get your genome sequenced, and then through various bioinformatic programs, you'll figure out, okay, which biosynthetic gene cluster am I interested in? And so you'll go to build up a plasmid, basically, where you'll put your gene and it'll be specific to which expression system that you want. Now, there's various expression systems available. There's not just E. coli, but there's also yeast, there's various fungi, and even up to flies to a certain extent. It depends on how complex your genes are. And so you'll begin the engineering process to optimize to your host expression system. And then you just let it culture and let it up the production. And then afterwards, you can basically do an extraction from that culture and then be able to analyze the metabolites from there. Now, my lab has been trying, and sadly, we haven't been getting there yet. But there has been one recent report actually of where a company in California was able to express lecanoic acid through this heterologous expression system. So nice results coming out there. So my PhD work was mostly, at least with COVID, couldn't go to the lab really all that much. So I had to figure out what can I do, at least for my project, linking genes to molecules. And so I've been running through different Cladonia species that are on GenBank. And there was actually this really interesting one, Cladonia rangiferina, as you can see on the screen. It's very pretty. I feel like I've seen this in Manitoba walking around, but it's pretty common in the US. It's also known as the Reindeer Lichen. And the chemistry for this lichen is basically atranorin and fumar protocitric acid. This is a little bit of foreshadowing. So I was running through and I noticed, hey, there is this interesting gene cluster. And this gene cluster actually was very similar to the arsenic acid gene cluster that my lab uploaded. And so I was kind of like saving that to the side, kind of just going like, I'm just gonna put this right here, put it on hold, work through these genomes. And then August of last year, there was this group in New York, they're part of the New York Botanical Gardens, sent an email to my boss saying like, hey, do you guys see this arsenic acid gene cluster too? Because we noticed this with our rangiferina samples. I was like, yeah, I did notice that. So we've been collaborating together and actually going through various Cladonia rangiferina samples, not only from the US, not only from Canada, but also from Japan too. So it's very interesting seeing how like we were able to recover this arsenic acid gene cluster from like all over the world basically. So we were kind of wondering like, okay, we're seeing it genomically, but where is it like is there actual metabolite production going on? And so we were doing various analytical techniques, TLC being like the least specific, but the one that is cheap and everyone can use. Wasn't there. HPLC, a little bit more sensitive, still wasn't there. But then again, there were so many metabolites that it could have just been hiding. So we went into high resolution mass spec and we were able to recover arsenic acid. So basically this is one of the samples from New York, and 343 is the arsenic acid peak or the arsenic acid mass. And so once I was looking at these various samples and actually able to see arsenic acid in some of them, I made sure to check on the fragmentation patterns because got to double check everything. And so we were actually able to see arsenic acid, which is really cool. So what we kind of gathered from this right now is it's really new results because we're still working on this paper. But basically, yeah, like Cladonia rangiferina has it, but it's making it at such a slow rate that like is not even existent. So now the hypothesis is kind of like, why is it so low? Is there a reason why this is so low compared to Uncialis, which is like obviously there? Or like, is there maybe something like evolutionary about it that it doesn't need arsenic acid so much compared to atranorin or protocitric acid? And so it's kind of like more questions now than ever, which is great because I'm only in my second year. But also like additional questions is like, can we apply this to other biosynthetic gene clusters that are out there? for lichens, and could we actually be able to achieve heterologous expression now that we at least seen one success story from it? So I would like to thank my group and like to thank my funding agencies and like to thank MSA for inviting me and Tanya for inviting me. And so now the floor is open basically.
Awesome, that's super cool. So I guess I have a question. I've been trying to understand and just wrap my head around metabolomic analysis, right? Because that also has to do with LC
Yeah.
mass spec. And so is there any recommendations you have? Are there particular programs they use to help you? Because like I'm not a chemist and and it's like I look at the stuff and I'm just like, all right, I don't know what any of this means. So is there any place that you recommend that we start looking into for this type of analysis?
I guess it depends on like, do you have a mass spec nearby to like be able to do these analyses on? Like, do you know like any groups to even like go off on? And I actually figured out using social media. Reddit has this like really nice mass spec subreddit that they've actually started putting resources together for anything mass spec related. But I would say definitely webinars are another great place to go to because I've learned a lot there also.
Interesting. That's so funny. Yeah, I did do some like metabolomic analysis. So we had a mass spec, we ran it through, and it was just like the person who was helping us was super helpful, and he's just like, all right, so we did it like it wasn't like specific, it was sort of like everything with everything. And we're just kind of like, all right, what are you looking for? I'm like, I don't know, what's cool?
That's usually the first question. Do you know what you're looking for?
Yeah, and so we ended up finding some things because we had the genome for the organism, and so I ran anti-smash, and I did see some particular things that like were super cool, and we're like, okay, do we see this? Does it match? And so we were able to find some of these things, but yeah, I was just like, okay, if you have any sort of direction or help or guidance, like that'd be really great. But yeah,
I just know what you're looking for.
Yeah, and just read it, I guess.
Reddit's nice.
Yeah. All right, so I see Julian has a question for you. Julian, did you want to ask, or did you want to, or I can read it for you?
Sure, I can ask. So I saw that you're primarily expressing your proteins in bacteria. Does that possibly cause problems trying to express the fungal proteins? I know some people do like Nicotiana or yeast or something like that.
So we were actually using Aspergillus oryzae. That was the PhD students before me, but we have one student right now who's trying to do the oxidative enzyme on E. coli. But yeah, like there's something about at least Aspergillus oryzae that just won't allow the expression to work. But we have seen yeast working a little bit better than that. So.
Okay, cool. Thank you.
All right, another question by Erin. Erin, did you want to ask yourself?
So usic acid is usually also associated with antibacterial activity.
Yes.
Do you think it's trying to use it in Cladonia as sort of a selecting for good bacteria versus bad bacteria sort of thing?
That's a possibility, but like, you know, it's interesting how Uncialis has like obvious quantities of arsenic acid just there, whereas Rangiferina is just like, I just don't need that much. So
I'm like, maybe it's like just a little bit. Just a little bit. I don't know.
Yeah, that's literally all it is just a little bit.
Awesome. Thank you.
Okay, thank you. And then Gulnara, looks like you have a question.
Hey, let's go.
Yeah, thanks. Great talk, really insightful. So I was wondering, like I know from looking at other genomes of Lichen fungi that many of them have lots and lots of secondary metabolism gene clusters that are like not linked to any compound or anything like that. And so I saw in the literature that they were considered like silent, that people were arguing that maybe they're not even expressed at all. So do you expect that all of them actually produce something? It's just maybe in like so low quantities that we didn't notice?
There's that theory, but also there's kind of like the theory of maybe the enzymes are just not functional is another possibility too, that just haven't been kicked off of the genome yet. So
Okay.
I think it's one of those two.
Yeah, no, that makes sense. Thanks.
Right. Do we have any more questions for Susan or maybe even Gulnara too? Yeah, all right. All right, Jorge Guzman has a question. Would you like to unmute yourself and ask, or I can read it for you? Okay, I'll read it. Okay, no problem. So Jorge is asking, any complete metabolomic pathway has been cloning and transferred in a bacteria? So I guess he's asking if, has any of this been done and transferred in a bacteria? And I guess, Susan, I know that you were kind of working on that with E. coli.
So that was only the cytochrome P450. I can say from reading the literature from the California group that it was actually producing the whole lichenic acid before actually breaking down first. So it's dependent on the environment. But it has been done, at least for yeast.
Okay. All right, well, thank you, Jorge. Okay, Julian, again, one more time. Would you like to?
Yeah,
all right.
I was just kind of curious about why are they making lichenic acid? What are they planning to do with it? I've used it as a dye, but kind of curious if the uses.
I mean, that's one of the more like not been really that studied that all too much, but I think they were just trying to say, hey, we've at least done something, and it made my boss pretty mad about that, or actually frustrated because he wanted to be the first group to do it. So
cool.
That's fair. Like you think in this realm you won't get scooped, right? Compared to like something medical. Yeah. All right, well, any other questions? I don't see any. But if there are any questions, I did post both Gulnara and Susan's Twitters, so you're welcome to message them there. And I kind of want to do like one little spiel before we end. So I am part of the MSA SPS, and we are having our nominations for the next year for 2021 and 2022 for our executive board. So we have all positions open. I did post in the Google form, but I can post it here again if you came in a little bit later. Please consider, it's a wonderful opportunity. Get to meet really great people, and the communication and just having the opportunity to work with other mycologists across the world. So yes, Maria. Oh, Maria, you're here. Welcome. Maria is the webmaster. Hopefully she'll stay next year too.
I think so. I thought that I submitted the application last week, and then I read your email today and I was like, nope, I didn't submit it.
Oh. No worries, whenever you have the time. All right, well, this was wonderful. Thank you again to both Gulnara and Susan for joining us today as awesome Lichen ladies. And tune in for our 2 speakers for next month. All right, thank you.